Hi, it's Ms. Bastido here. We're going to be covering Unit 3, Topic 1, Describing Biodiversity. This might be a little bit of a long video, but I'll try to get through it as quickly as possible, but still making sure that you understand what's going on. So we'll kind of just jump right in. These are the dot points from the syllabus for this section. We'll mostly do it in order, although there might be a little bit of jumping around in a few spots. And so we'll really be talking about species diversity and ecosystem diversity. We'll spend time talking about different measures of diversity and how we can use those measures along with species interactions and abiotic factors to compare different ecosystems, either over time or maybe in different areas. And then lastly, we will talk about how environmental factors can limit distribution of species and the abundance of species in an ecosystem. And so in order to really talk about ecosystem, there's a few terms that you really need to know. While these aren't explicitly stated that you have to define them in the syllabus, they certainly will be used, and knowing them will be useful to you and in being able to talk about these things intelligently. So many of these things you've probably defined before in other science classes before you got to senior biology. And so this section is over biodiversity, but really we're talking about ecology here in general, and ecology is the study of relationships between species and their environment. So that's those interactions that occur between the living and the non-living. And so ecology would be studying all of those different kinds of relationships that occur. Obviously, a habitat's a place where an organism lives. You probably already know that. Differences between populations and communities. Populations are groups living in the same area that are the same species, whereas communities are groups living in or different species living together in the same place. And so you could have a population of wallabies, but then the community would also include all of the plants and the like in this picture here, the little crabs on the beach and things like that. That would all be part of the same community. And then the ecosystem is all of those living things, the community plus the abiotic factors, which are the non-living things, which I've kind of written here. So abiotic means non-living, biotic means living. And then lastly, we'll talk about this actually a bit later in the curriculum, but a niche is a role an organism plays in the ecosystem. So it includes all of its interactions, where it lives, what it eats, what eats it, if there's any symbiotic relationships that it might have, and that would be a niche. And we will talk about niche a little bit later on, not so much in this video. And so then to define biodiversity, we would say it's the sum of all the variation. This includes genetic variation, species variation, and ecosystem variations. That's the way that we'll sort of define biodiversity. Biodiversity is important because biodiversity is what makes ecosystems stable. The interactions that occur within an ecosystem is important. If biodiversity is low, those species interactions could be severely impacted. Lots of cultures really rely on biodiversity. Indigenous cultures rely on biodiversity. Lastly, tourism and economics, obviously, especially here in Australia, we know that tourism to the Great Barrier Reef or to the Daintree is really important. And that's due to maintaining that biodiversity. So not maintaining it would have not just biological impacts, but also economic ones as well. And so one of the ways that sort of things that you need to think about is that organisms, we need to know that species, the way in which they're scattered in an area is their distribution. So how are they distributed over an area? And then their abundance is the number that there are in that area. So the distribution is more influenced by abiotic factors, whereas the abundance is more influenced by biotic factors. And we'll talk a little bit more about distribution, but we really, when we're talking about distribution, often talking about across a gradient of some sort. Abundance is just the total number of organisms in that area. And so distribution of a species can vary depending on the type of species. So some species are uniformly distributed across an area. Some are randomly distributed across an area. And then some species really have clumped distribution. And so the distribution of a species has to be considered when you're determining what method of sampling that you might do. And we'll talk about that a little bit later. But if we have clump distribution, we really have to think about how we sample this whole area. If we took too many samples from over here, we wouldn't see any species, and that wouldn't be very representative of what was true. So thinking about how species are distributed. And then different things will limit the abundance of a species. So you could have chemical factors, some kind of abiotic factors like soil types, temperature, climate, rainfall, all of those things can limit the abundance of a species. Competition, either between the same species or between different species, both of those could impact the abundance or how many species there were, or how many individuals there were. Disease can obviously lower abundance if there's a spread of a disease. And then exploitation, such as like an invasive species comes in and creates a pressure on a species that could lower their abundance as well. And so some of these are density dependent limiting factors and some are density independent. Density independent would be sort of more of these and maybe the exploitation, although that could be with density dependent as well. And then these would be density dependent. Okay, so we've talked a little bit about this in class already, but we can measure diversity in different ways. And depending on what we're looking at, one measure might be better to use than another. But all of these are legitimate ways to measure diversity of an ecosystem. So we could look at percent coverage. This is especially useful if we're talking about plant cover, or we wanted to know even shade cover from trees. Percent coverage is an important measure of diversity. And that we'll look at an example of that in a minute. Species richness is simply the number of species present at the site. And so that would be species richness. Species evenness would be like how evenly distributed is each species that is present. So if there's 4 species present in one site, but there are 10 of one of those 4 species and only 1 of the other 3 species, then they're not very even. But if there was 10 of each of the 4 species, now we have more evenness in terms of the whole population, the whole community being even. And then Simpson Diversity Index, we'll talk about that in a minute, but that takes into consideration both the species richness and the species evenness. And then lastly, we can calculate percent frequency. So how often do we see the species when we sample? So if you had 10 measurements and we found them in 6 out of the 10 sites, then we would say 60% would be the percent frequency. So here's an example of percent cover and species richness. So when we look here, we have 2 different plants and they're labeled species A and species B. So our species richness here is 2. We have species A and species B. Our percent coverage, we're using here a quadrat, and so we're trying to, it says each square is 4% of the quadrat. So we're trying to see how many squares does this one take up. And so we could say approximately that it takes up 5 squares. So there's like definitely covers all of that one and that one and that one, about half, and that one about half, and about half of that one and a bit of that one. So we're really estimating here, but about 5 squares. And so we'd say about 20%. So we say 5 times 4, and that's about 20%. This one here maybe is only about maybe 8% or 4%, because it looks like about 1 square. And so that's how we would estimate the percent cover. You can see how that could be a bit of an inaccuracy there, but mostly we can If we have these smaller quadrats and we know what the percent, if that one's fully covered, then we can make a pretty good estimation about the percent coverage. Here we have an example of comparing two communities, and we look at the species richness and the species evenness. So how many species are found at each site? So here we have, there's one species here, here's another one, and then another one, so we're at 3. This one here, the hippo's 4, these gazelles are 5, and then these are down here are 6. So there's 6 species in community 1. If you look at community 2, there's 1, the lions is 2, giraffes is 3, hippos 4, 5, and 6. And so our species richness here, both community 1 and community 2, is 6. However, the evenness is quite different. And so you can see community 1 has a dominant species of elephants. And this community has a much better distribution. It looks like there's 3, 4, or 5 of each species. And so this is a much better distributed community, and so it has better evenness. Higher evenness, this has much lower evenness. Another way to look at percent coverage, if we were using a transect, which we'll talk about a little bit later, if we were using a transect, we can say that this one, this species here, the percent of grass cover, so this is the grass, I guess you could say, it goes from 1 meter or 1.1 to 2.1 or something like that. And that's about 1 meter. And then this one's 1.7 and this one's 0.6 and this is 0.8. So we could add all that up and then divide by the total of the transect. And so we said 1 plus 1.7 plus 0.6 plus 0.8 divided by 10. And that gives me a 41% coverage along this line. And then for the other graphs, we would say, so for this one, you could practice this on your own, but you'd say 0.9 plus 1.3, and then you'd divide it by that 10, and that would tell you the coverage of this plant. This is just another way to do it. And oftentimes we'll do this when we are looking at trees and we're looking at percent coverage of the tree, in other words, shade coverage of the tree. And so we can oftentimes do this. When we calculate Simpson diversity index, you'll be given the equation. It could be on your data test, it could be on the external exam. And so remember that it takes into account both species richness and species evenness. Mistakes are made by students when they forget to subtract from 1, so don't forget that part of the equation. And then the other part is they just aren't paying attention to what these letters mean, nor occasionally they don't know what this means. So this is a sigma, and that mathematically means the sum of. So we're looking at the sum of lowercase n times n lowercase n minus 1, and then n, capital N minus 1. So the little n is the total number of individuals of each species, and I'll show you that in just a minute. But the capital N is the total number of individuals of all species. So if we were to look at an example of this, here you can see this is my lowercase n, the number of individuals of sea couch grass. So that's 21. N minus 1, so 21 minus 1 is 20. So then we take 21 times 20, and we get 420. Remember that we have to do that with each of these species. Then we want to find the sum of little n, n minus 1. So we say 420 plus 0 plus 56 plus 0 plus 132, and 608 would be our sum here. We also need to know what are the total number of individuals, and that's when we count up all the n's. So we add these up to get to our capital N, which was 43. Now, for location B, we do the same exact thing. We ended up with 520 for the sum of n and minus 1, and we ended up with lowercase n, for capital N, we ended up with 47 species, or individuals. Keep in mind that when we look here, our species richness is 5 for both. There are 5 species found at both sites. And so here is the math. Here we say 1 minus 608 divided by 43. 43 for location A was capital N. And we say capital N times capital N minus 1. So it's 43 times 42. So what's 1 minus 608 over 1806? And then we end up with a diversity index of 0.663. We do the same thing for location B. We end up with 0.759. Really useful here when you're comparing two sites, because we can say that the diversity at location B, according to the Simpson diversity index, is higher than that of location A, indicating it has higher biodiversity. You'll always get a number between 0 and 1, 1 being the highest diversity and 0 obviously being the lowest. And so oftentimes you can compare these two. On a data test, just be careful if they tell you to go to 2 decimal places that you do that, or 3 decimal places, then you do that. So when we're looking at, those are the different types, ways of measuring diversity, but we have to be able to sample a population, a community, to be able to figure those out. And so there's 3 types of sampling. We're really only going to focus on 1 or 2 here. We will talk a little bit about stratified, but maybe get into that a bit more later as well. So random sampling, it is just like it sounds, which is you randomly select areas in the area that you're trying to sample from, and you randomly select spots within that area, and you can use a random number generator to do that. Systematic sampling means that you are going to, in this case, this is a quadrat sampling, so I'm going to go every 4th quadrat and sample. That is systematic sampling. Or if you're along a transect, you might go every 4 meters you're going to sample. That is systematic. Stratified sampling is when we see that there's a difference in strata. So a lot of times what you'll see is you would be looking at an area and maybe 70% of it is really muddy and 30% is sandy. And you want to know the distribution of, or the abundance of a certain plant species in that area. So if you want a really good interpretation of the area, if 70% of the area is in the mud, then you can't just use random sampling. You really need to, 70% of your samples need to come from the muddy area, and 30% of your samples would come from the sandy area. So kind of an example here, but if this was my population, I would want 50% of my samples to come from this area, because this area has some certain strata that is different from the other areas, and 50% of the area is like that. And so this is called stratified sampling. You really have to understand the distribution of the strata to be able to do stratified sampling, though. Our sampling techniques that we will really use in this class would be quadrats, transects, and belt transects. There are other sampling techniques, but these are the ones that we'll probably focus on the most. So with a quadrat, we take a square, we separate it up into smaller squares, and we can determine percent coverage. This is really nice for determining the abundance of a species, how much of this species is really present. It's not really good for, at least alone, it's not really good for figuring out distribution along a gradient. And so we wouldn't really be able to do that using a quadrat. So in this case, this is what we've already done before, is we could make an estimate of what percent this oval covered. And so we know it's 1 full square here, and you could then estimate the percentage, and you could do that for each of these. A transect is a little bit different. So on a point transect, what we have is it's showing distribution of a species along a gradient. A good example, we usually take a transect and move it away from an abiotic factor. And so an abiotic factor could be a lake or a river, or it could be a lot of things. It could be a path or a trail on a hiking track or something like that, or on a bushwalk. So we would be taking a a transect away from that site. And we would really be looking at how does the species change as we move away from that site. So, you could do this on a beach. You could do it from from high tide to low tide. Like, how does the species change as we move along this transect? So we would only count the species that would be touching the line. And so we would not be able to determine abundance from something like this. Now, it is important to know you would have more than one transect because you would do more than one trial. However, you would only count species that were on the line, actually touching the line in a point transect. And so this is a random sampling along a transect. You could also do systematic sampling, which is often done along a transect, because if you're really trying to figure out how does it change as it moves away from the water or this lake, if you're looking at how do the tree species change as they move away. So then we would really see, we might see maybe there are no trees as we're closer to this lake, and then as we move out, the trees get bigger and bigger and bigger. That could be something that we would see, or maybe the species change as you move away. Again, we would not be able to look at the abundance. We'd just be looking at distribution. But you could do more than one trial, so you would have a good idea of the distribution after doing many trials of that. Lastly, you can do a belt transect. So belt transects are useful because we can show not only the distribution along the transect, but also the abundance. And so oftentimes what happens is we put down a transect and then we use quadrats, and we would, this one only has one quadrat, but we usually go out either in both directions, but between two lines, and we would have a lot of sampling to do. This takes a long time, and it can be damaging to the environment depending on how much you would be destroying the environment trying to walk on it and to collect these samples. But it does give us a much better idea about the distribution and the abundance of the species. So we would learn the distribution along the transect, but then we would also be able to tell how many species there were and what was the abundance of the species, because we were doing a lot larger amount of sampling with the quadrats along with the transect. Something to keep in mind is that when we're sampling species that are moving, we can't really just use a quadrat. Just using a quadrat and I'm trying to sample fish, there's going to be no fish in the quadrat because, well, they move. So there are different methods for capturing species. So if we were, an example of these are pitfall traps or Burley's funnels, but essentially you're just trying to capture insects in this case, in the Burley's funnel, and capturing those insects, and then you could make some conclusions about where those were. I think I did this in my uni course with ants. We built some kind of something like a Burley's funnel for that, and to figure out how the ant species changed as we went from the grassy area all the way into the forest. And so you could kind of see how they had changed. But we did systematic sampling, and we put out the Burley's funnels along a gradient, across the gradient. And, instead of using quadrats, because obviously with ants, we were not going to see ants in the quadrats because they would be moving. And so this is just to be thinking about that a lot of those other using quadrats is really for plants or species that are more, we call sessile, meaning that they don't move. When we're talking about mobile animals, we need to think about other things. So with fish, it would obviously be different and might be a mark recapture, which we'll talk about a little later, to estimate population size. And so the way that we could do that, and by a little later, I mean now, because I forgot I put this slide in there. We can do that by doing the Lincoln Index. And the Lincoln Index is going to come back later to us when we talk about populations. It's a way of estimating the size of a population. And so you can do a capture recapture. So what you really do here is you do an initial capture of a species, you mark them, and then you release them. And then you come back after a little while and you do another capture. And of the ones that you capture, keep note of the ones that were already marked and the ones that weren't marked. And so if they're not marked, then you haven't caught them before. And so we use this equation to determine that. So sometimes it's not really, sometimes it's referred to as N1, N2, N3. But the originally marked ones are here. This is the total number in the second capture. And the lowercase m is the number that you recaptured here. You divide the number that were marked in the recapture. Let me repeat that. That was a bit confusing. So the capital M is the number that we captured the first time. The lowercase n is the number that we captured the second time. And then we divide it by the number in that second recapture that had a mark on them from the first time. So let's say I mark 10, I release them. And then the second time I captured 10 again. So I put 10 times 10. And then of those 10, 5 of them had a mark on them. So I'd say 10 times 10, and then I would divide it by 5. And that would tell me approximately the size of the population. So I'd say 10 times 10, which is 100, divide that by 5, and that gives me a population size of about 20. And so that's just a way of estimating the population size. Again, we'll talk about that a little bit later, but sometimes a mark recapture or something like that has to be done for species that are moving. So we won't necessarily use quadrats and that sort of thing. I apologize that this video was a little bit long, but hopefully it covered everything so that you could understand a little bit better about biodiversity and sampling methods, because we'll be using a lot of these in our IA2 and in some pracs coming up that we have in class. Let me know if you have any questions.